turnout for the first seminar of the term, which I think just shows the value of the speaker we have today and the topic. So I'm delighted to introduce Tobias Reichenbach, who's come from Germany. He's currently employed at the University of Erlangen-Nuremberg, and he works in the Department of Artificial Intelligence. His main interests are really the neurophysiology of speech now, but he was originally trained as a physicist. He worked as the BA and Masters in Leipzig in Germany. Then he moved PhD to Munich, and then he went to New York and studied at the Rockefeller Institute as a postdoc. And then he set up his own group at Imperial College London, where he really got into speech, I think. That's when I first really began to follow his work. And now he has his own lab in Germany, where, as you'll see, he's doing some really exciting stuff. So we're really looking forward to your talk. Sorry, somebody lost a bike key. It was found at reception. So I'm just going to leave it here. And if you think it's your key, you can just sneak forward. We're really looking forward to your talk, which is Decoding the Neural Processing of Speech. And there are seats at the front, guys, if you want.
Well, thanks very much. I'm humbled by this turnout. It's nice to see such a full seminar room. So I hope I can live up to the expectations, of course. Also, I should say that I cover quite some different works that we've done over the years. So some things might be a bit fast or quickly summarized. So I see a lot of young faces in the audience. That's great. Don't hold back if you have questions, right? I think this is mostly also for you. Also, of course, for the older scientists who have all the experience and know everything anyways already. But it's especially for you guys who are still young and you guys want to learn. So don't hold back. So let me start by just pointing out what my group is working on. We call it sensory neuroengineering. The idea is that we look at our different senses. Most of it is hearing, as you know already. But in principle, we have 5 senses: touch, smell, vision, hearing, and taste. And the goals that we have is to, on the one hand, do foundational research to understand the neurobiology of multisensing. Then second, to diagnose impairments with sensory processing. And third, to try to restore sensory impairments when they happen. Now, as Usha already said, my work is really mostly on speech processing and, in a bit larger sense, on hearing and communication disorders. Where the important, it turns out that there's a high prevalence of such disorders. So hearing and communication disorders affect about 20% of people worldwide, and already in children, 5%. So not as many as in the general population. Certainly hearing problems, communication problems increase as we age, but already in children there's some prevalence, and Usha knows about quite a few of them and is doing great work to find out what's going on there. There are several disorders, several types. This is an overview of the auditory system. We see that we have sound coming in the inner ear or cochlea. That's where the mechanical sound signals are transduced into electrical signals in nerve fibers that are then forwarded to the brain. So that goes then here along these nerve fibers to the so-called subcortical brain areas, brainstem, midbrain, inferior colliculus is probably an important station that I'm gonna talk about again later a little bit. And then the auditory cortex, primary, secondary, other areas of cortex, where certainly most of the heavy lifting, so to speak, in terms of sound processing happens. So where we look at whom do we want to attend, how do we extract meaning from these pressure fluctuations that we perceive. And all along these stages, there are sensory impairments that can happen. So we know a lot about impairments in the inner ear, that is called sensory neural hearing loss. So when so-called specialized hair cells that normally transduce the pressure fluctuations into the electrical signals, when these fail. but we can then also have issues with the forwarding of the electrical signals and hair cells to the auditory nerve fibers. That is called sometimes hidden hearing loss or cochlear neuropathy. Then we can also have difficulties that arise later in the auditory system to do with the processing in the cortex, that is auditory processing disorder. Also language disorder, that is then really issues with kind of interpreting these signals that arrive in terms of linguistics, semantic, syntactic things, and so on. Now, in my group, we look a lot at the so-called cocktail party effect. I could almost have done maybe a photo from the tea room where we just were, because that was really a real-life demo, right? I mean, we had a lot of people, which is great, but that means also a lot of people talking at the same time. And that's a difficult listening environment. So I don't know if you realize that. Maybe some of you, I'm sure, did. That it's not easy actually to understand that other person with all this background conversation. Same here in this picture, with all these conversations going around, it's quite difficult actually to attend to one particular person. Now, as a healthy younger person, we typically still do it very well, but for somebody who has hearing difficulties, that becomes much more difficult. And that's something that we are interested in. On the one hand, how does our brain do it? How does it accomplish it? But then also what goes wrong when people have difficulties and how can we potentially do something about it? And that's kind of one thing that I want to tell you about, some work that we are working towards, more long-term goal, nothing that we can solve in the next couple of years, but we hope we can make some progress towards that. And that is called, what we call smart hearing aids. What is the idea here? Well, if you have a normal hearing aid, then the difficulty with the situation that I just mentioned is that it amplifies all the sound in the surrounding, but not kind of preferentially a particular person that you want to listen to. It also has many settings that have to do with different frequency bands, certain gains in the hearing aid, that are difficult to really fine-tune precisely to a particular hearing loss or to a particular situation, less noise, more noise from different sources that you might find yourself in. And the outcome, unfortunately, is that people wearing hearing aids can use them well in kind of simple acoustic situations when you've got a single speaker, perhaps like now, but much less so when you've got, again, these different competing speakers or sound sources that happen at the same time. Now, what is our vision of a smart hearing aid? The vision of the smart hearing aid is that it should somehow automatically detect the focus of attention, so figure out who is, say, a particular speaker that you want to listen to, and somehow selectively amplify that, reduce background noise to make it easier for you to focus on that particular voice. Ideally, such a smart hearing aid would also, in some automated way, fit all these myriad settings that this hearing aid possesses to your particular hearing loss, and maybe also adaptively so, depending on the environment that you're in. Now, how do we think we might get there? And again, that's more in the medium to long term. But we work on approaches that use brain recordings, namely through EEG. So EEG means electroencephalography. So we measure brain activity from electrodes that in this case sit somewhere in the outer ear and in the ear canal, or perhaps from these devices that wrap around the ear, around both ears, and can measure some aspects of brain activity from these so-called scalp electrodes. I should say a lot of work that we do, what I will show you later, is done using full scalp EEG. So then we have a lot more sensors that kind of cover almost the full head as much as we can. But later, our goal with the smart hearing aids would be to get as much information as we can out of these simpler wearable devices. What do we want to do with this with these recordings? Well, we want to, for example, detect what is the focus of attention. So if you have several competing sound sources, then figure out which signal is somebody listening to. And also decode speech comprehension. That means ideally pick out if somebody wants to attend to that speaker, but somehow fails understanding them. Because that would then be a signal that we need to change some settings, we need to change something to again improve speech comprehension, and then ideally verify that again in this automated way. How do we think that we can achieve that? Well, our general setup is that we want to look at neural responses to speech, again as measured from EEG, and extract useful information about behavior being attention to speech, or speech comprehension, or maybe other things, or maybe listening effort, things like that. And that's a kind of general outline in a few steps how we might get there. So we want to use artificial intelligence to do this by studying both the encoding of certain speech features in the brain. That means how does the brain represent speech, at least to some degree. And then as another step, decode neural responses to particular speech features that we find that the brain uses, that it encodes. And then third, importantly, relate these decoded responses to behavior. So again, things like attention to speech or speech comprehension. So I now want to kind of, starting from this general setup, dive a bit in to give you an example of a particular type of brain response. It's not the only one, but one particular type that we think can help to do that, that we've worked on quite a bit. And these are brainstem responses. Brainstem responses means these responses come from lower parts of the brain, so not yet the auditory cortex, where the higher level processing happens, but more these low level parts. So you might wonder, well, if we want to look at behavior, things like attention or comprehension, then why do we look at brainstem, where more this low level acoustic processing happens? But it turns out that there are actually feedback loops that go from these higher level areas, the cortex, back to the brainstem, and even all the way back to the inner ear. So the auditory system in reality is not just this feed-forward system that I had initially on one of my first slides, but actually a closed loop system where also output from higher level processing centers like the auditory cortex somehow feeds back to lower level areas. And a lot about what this does is not well understood. And that's also what we wanted to look at kind of from this basic science sense of view, namely what is that doing? Are these low level areas already involved in things like attention? And then if so, could we maybe also use that for some of these practical purposes? Now here is an example of a brainstem response. This is a single vowel R, and we see here a so-called spectrogram. Now, so spectrogram means we follow over time, in this case 100, 200 milliseconds. We look at what is the spectral content, that means which frequencies are present at any particular time point. And we see a very particular structure, namely we have a so-called fundamental frequency here, maybe 100 hertz, and then multiples of that. So 200 hertz, 300 hertz, 400 hertz, and so on. So that is typical of so-called voiced parts of speech, that they contain this fundamental frequency that arises from opening and closing of the glottis and all the higher harmonics. And we see then in the brainstem response that we measure from many repetitions of this vowel a very similar structure, not quite the same. So we see here the highest amplitude is at the fundamental frequency, say roughly 100 hertz, and then a bit less also at the higher harmonics, but really most at this fundamental frequency. And the reason is that the nervous system acts a bit like a low-pass filter. That means it can barely respond at high frequencies like 1 kilohertz, but still to some degree at 100 hertz, although later most of it actually at yet lower frequencies like rather 1 to 20 hertz. But to some degree, especially at the brainstem, still also at frequencies like 100 hertz or even higher. Now, we wanted to look at, can we see this brainstem response when we have continuous speech? So not these vowels like a single vowel R that is repeated like a few hundred times, but rather somebody listening to audiobooks, and we rather then do some statistics, use some AI in other words, to see from the recordings, can we find signatures of these responses at this fundamental frequency. And that's work that was done by a postdoc in my group at the time. He's now a lecturer at King's College, has his own group. And we looked at starting at spectrograms from natural speech. There we see again the structure with the fundamental frequency and higher harmonics. But we see also that this fundamental frequency varies. Because when we ask a question, we might raise our voice. If we want to emphasize something, we kind of change the pitch. And so that leads to these variations in fundamental frequency. But that's our difficulty, because how can we now relate this to these EG recordings that we do simultaneously? Our approach was that we extract something that we call fundamental waveform. So here, black is the actual speech signal. That's a complex signal. There are a lot of frequency bands involved, going from somewhere like 20 hertz up to 10 kilohertz, perhaps even 20 kilohertz. The fundamental waveform is a much simpler signal. You can basically think of it as a bandpass filtered signal in the range of the fundamental frequency. That means that each time instance, this red signal fluctuates at this fundamental frequency, which changes. Sometimes it's higher, sometimes lower, but always, so to speak, in sync with what the fundamental frequency is at that given time point. And then we can take this feature, this particular feature of speech, this fundamental waveform, and relate it to EG recordings. Now, what you have to know about EG recordings is that they're very noisy. So it's a very poor measure in a way of brain activity, because we don't put an electrode into the brain and record from single electrodes, but it's really a coarse-grained measure of a loads of activity from millions and millions of neurons. So we need statistical approaches. In this case, we use methods like, for example, linear regression. That would mean a linear predictive model to either, in a forward model, predict the EG recordings from the fundamental waveform, or we can use a so-called backward model. That means we reconstruct the speech feature, in that case, the fundamental waveform from the EG. Or there's also some in-between way called canonical correlation analysis, where one tries to find an intermediate space in which these two types of signals, the acoustic one and the EG one, in some sense maximally correlate. So I show you here examples from a so-called backward model, where we reconstruct the speech feature from the EG, because that's got advantages when it comes later to analyzing effects of behavior like attention. And you can see here results from AT in subjects and also from a population average. The white bars denote the reconstruction, and what is measured here is the correlation between the reconstructed speech envelope, not speech envelope, but the reconstructed fundamental waveform and the real fundamental waveform. And gray is a noise model that we get through certain means, just to check what we get by chance, and then seeing whether the thing that we get from the real model is that better than chance. Then we say we got an actual measurement. Now you can see that these correlation coefficients are quite low. They are like maximally here, maybe 0.1, or for one subject, maybe a little bit higher, 0.15. Minimally is something like 0.05. So these correlation coefficients are normalized. So 1 would mean that there's perfect correspondence between the two signals. -1 would mean just with a minus sign. 0 would mean no correlation. So correlation coefficient of 0.1 means, yes, there's some correspondence, but also quite little. Now, we don't think that's surprising, because, of course, we reconstruct this thing from the EG, but of course the EG signal contains all kind of activity. So this brain stem signal that we see is just a small part of that. And of course there's all kind of other stuff in that, which means that we won't get this perfect reconstruction. And that's why this comparison to these noise models is important, because we see here for these first 3 subjects, this is not significant. So in that case, we don't get a significant measurement. In the other cases, yes, that we do get a significant measurement, but we need the noise model really to assess whether these relatively small correlation coefficients that we obtain, whether these are significant or not. And when we have this backward model, we can then look at characteristics. So I should say that what we include in the backward model are coefficients for every EG channel and for particular delays. So in this case, delays between, say, -50 milliseconds extending to 150 milliseconds. so we basically ask for the EEG channel, if you have a certain delay, does that help to explain this acoustic feature, this fundamental waveform? And it turns out that the delay that mostly helps is 8 milliseconds. And that basically tells us that there's a latency between the acoustic signal and the brain signal of about 8 milliseconds, which fits with previous measures of brain stem responses. That's in the auditory world a very short delay, but again, the brain stem is very close to the ear, so it doesn't take long to get there. So that's a confirmation in a way that we measure really a meaningful brainstem signal. And we also can look at so-called topographic maps. That means we look at where, basically, if you look at the coefficients at 8 milliseconds at the different EEG channels, which ones give a high magnitude and what are their phases. And we see that the highest magnitude come from the so-called mustard electrodes that sit basically below the outer ear or just at the lower end. A little bit here from CZ. And these are also 180 degrees out of phase, which is another confirmation of this brainstem source, because we know from other types of auditory brainstem responses that these basically induce like a dipole responses somewhere close to the ears and are best measured with electrodes that are more or less at the mastoids and somewhere near CZ. So we see the same picture emerging basically from the data found by this linear regression model that's been trained on all the data that's required here. Now, the question is always, where exactly do these responses come from? We don't have a precise answer. There are certainly different sites in the brain that contribute. One origin we think is in the inferior colliculus. We did some modeling work with another PhD student in the group, Marina Saiz Alina, where we used a model published by other people before of middle ear, inner ear, cochlear nuclei, inferior colliculus, and we can then model responses, again see, analyze it with our pipeline, and we see then the largest responses coming from this inferior colliculus that, as I mentioned before, is in the midbrain, kind of between the brainstem and then the primary auditory cortex. Now, okay, we have a method for measuring these brainstem responses to speech, but we wanted to see, as I said, do they actually encode? Do they do anything? Do they tell us anything about behavior? On the one hand, we wanted to see, again, is there this top-down feedback? Does that already work at that level? But then second, also, could it potentially be used for applications like these smart hearing aids? So we did a very classic experiment in auditory selective attention. Namely, we took two speakers, a male and a female one. We mixed them together so people listen over earphones to two speech signals at the same time. And we measure the neural response again through EEG. And we then analyze, we tell people to sometimes focus on the male speaker, sometimes focus on the female speaker, and then compare, do the responses look different depending on whether sometimes somebody attends the male voice or the female voice. And that's what we find. So here I show data from different subjects for listening, so brain stem responses to the fundamental waveform of the male voice. In dark blue when the male speaker is attended, and in light blue when the male speaker is ignored. And we see for almost all subjects, there's a stronger response when the male speaker is attended than when the male speaker is ignored. And we can do the same analysis for the female voice. Again, we compute the brainstem response to the fundamental waveform of the female speaker. And again, ask the question, how strong is that when the female speaker is attended compared to when that voice is ignored? And again, this dark red color, that's for the attended condition, this pink or light red color for the ignored condition. And we see again in most subjects a stronger response when the female voice is attended. So that is quite surprising. Initially, it was surprising because, again, that's an effect of attention that happens at that subcortical level, probably driven through this top-down feedback, but one that emerges very clearly in this data. We wanted then to go a step further and see, well, although the signal is weaker than other cortical responses, it happens at a fast timescale because it happens at somewhere like 100 hertz, depending on the fundamental frequency of that particular voice. So could we potentially use it to decode attention from short segments of speech? So real time, or somewhere, say, getting close to somewhere maybe like real timer. And I won't go through all that in detail. That's work by two other PhD students in the group, Octave Etat and Nikolai Kegler. We built different decoders to look at reconstructing this fundamental waveform for ignored attended male-female speakers. And it turns out that for the attended models that follow this green line, we then measure how precisely we can decode the locus of attention. Accuracy is given here from 50% to 100%. So 50% would be chance level. Actually, all that's marked in gray is chance level due to certain properties of the decoder. And we look at the accuracy as a function of the duration. That means how much data do we give? Is it just 1 second? Or is it 10 seconds? Or here up to 30 seconds? And we see that the accuracy is not high, right? It's not like 100%. It's maximally for 30 seconds, maybe like a bit above 70%. But it is higher than chance, and it stays higher than chance even for very short segments of just 1 second or so in duration. And what was also quite surprising for us, but maybe also again not, is that if you reduce the number of EEG channels from 64 that we had originally to just 3, meaning mastoids and CZ, then we find more or less the same picture. We more or less have the same decoding accuracies, again maybe 70% for a data segment that's 30 seconds long, going down to something like 1 second, going down to close to chance level then for 1 second, but still staying a bit above chance after that timer. So that's not yet real-time decoding, right? It's also not yet super precise, but I think it shows a bit the promise that this can be added as a method, maybe with other methods, other acoustic features, to get closer to this real-time attention decoding. And it can also help with related things. That was a so-called IEEE challenge, so a challenge by the Society for Electrical Engineering Information Processing, the ICAS conference on speech processing just this year by one of my students who is still at Imperial College, Mike Thornton. He looked at this challenge where it wasn't about attention decoding, but it was rather about so-called match-mismatch decoding. That means you were given 2 particular EEG segments and an example of speech. And one of the EEG segments was recorded while people listened to that particular speech, and the other one was from some other speech, and that was not given. So one EEG segment was matched to the speech, while the other wasn't, and the task was to decode which is which. So somehow find relations between EEG and speech, again, what we need also later for attention decoding. And there are certain features that we took as a baseline, namely, I'll come to that later, but to do with cortical responses that have to do with amplitude fluctuations in speech, so-called envelope tracking. And that is what we call here the baseline model. So that is a big part of how we can decode what is the actual EEG segment. But when we add what we call speech FFR, so basically these brainstem responses to pitch in speech, then this decoding accuracy improves further, and we can further get a better decoding. And so Mike actually, with this approach of adding these speech FFRs, these brainstem responses, won this EEG decoding challenge, which is a great success for him, of course. Now, this is some work that's fresh off the press, extending a bit further what we do on the speech FFR, namely, well, I said already, there are different contributors to that response in the brainstem, but it turns out that there's another contributor that I didn't mention yet, and that is cortical activities. So not just brainstem, so the way I wasn't quite honest with you so far. These cortical responses, however, can basically almost not be measured with EEG. They more or less only appear when we use MEG. So MEG is a different measurement modality where we measure the magnetic activity that is associated with the electrical activity in the neurons. We have fortunately a system for that in Erlangen. And Alina Schüller, one of my PhD students in Erlangen, she worked quite a bit on that. So she acquired MET responses where people listen again to two competing speakers. In this case, actually, we had two male speakers, just to have a bit different setting, with quite low fundamental frequencies. One still had on average a higher pitch than the other one, so we call them the high pitch speaker, the other one the low pitch speaker, just to differentiate with them. We then also compute, in this case, actually two acoustic features. One's the fundamental waveform that I mentioned, that's obtained by bandpass filtering the speech signal around the fundamental frequency. The other one is one that we call envelope modulation, that basically looks at the higher harmonics and looks at how much energy fluctuation is contained in the higher harmonics at the fundamental frequency. So again, related to the fundamental frequency, but representing another type, so to speak, of things that happen in the speech signal and that we think, that we know, can also drive neural responses at the fundamental frequency. And we find, again, quite strong effects of attention. First, we find where these cortical contributions happen. That had also been reported before by other people. We basically built the same models that I showed you before in terms of linear regression, looking at how we can explain brainism or cortical responses through these acoustic features at different time lags. And we see that the time lags that matter now are later. They're around 35 milliseconds, so no longer 8 milliseconds, which makes sense because, again, these being cortical contributions means there has to be another delay between brainstem, midbrain, and now on to the cortex. We can also source localize them, so they're actually, interestingly, more located in the right hemisphere. We also know that music processing often happens more in the right hemisphere. Maybe it's to do with that, but we see a very clear lateralization to the right hemisphere and more or less kind of auditory cortex regions. Here in this case, maybe a little bit towards primary auditory cortex, but I would say overall, that's difficult to say. That's quite still quite fuzzy at the moment, this picture, but certainly in this auditory cortex region. And I won't show you all the data, but again, here in this data set, the red line, that's for the attended signal, the black for the ignored. These are population averages. We also have data for single subjects. But for the population averages, very clearly, we see a much stronger signal for the attended case than for the ignored one. And we see it also to a large degree in single subjects. That's also interesting. So it shows kind of still also just going back to this particular response, how rich that picture is, and which might also be interesting for other studies, maybe as we discussed with Usha earlier, maybe on dyslexia. Maybe one could also pick out differences in these responses in children that have dyslexia or those that have not. All right, well, with that, I want to kind of go away from this first attention decoding part and go to another question, namely, once we have decoded attention, then how can we actually help that, say, if you think about the smart hearing aids, right? So if we had solved, we are not there yet, right? But say we had solved that problem, how can we that help people to better understand the target speaker? And I said, well, yes, we can reduce background noise in the hearing aid, and that is certainly one possibility, but it's also not that trivial. So people have worked on it for a long time, and it's surprisingly difficult actually to clean up such a sound signal well. And we therefore thought also, are there other possibilities? And that's where multi-sensing comes into the game. And we namely did some work on audiovisual speech comprehension. By audiovisual, well, our brains can integrate visual signals with auditory ones. So if you listen to me talking, then you see my moving lips, and that has some information about speech, and that makes it easier for you to understand me. Some people are expert lip readers. They can even understand people just by watching them without hearing anything at all. Most of others are kind of moderate lip readers. There's also some variability, but for all of us, it does make a difference. Now, unfortunately, we don't always have access to seeing somebody's face. I think maybe in a train station, maybe there's some announcements coming over the speakers, but at the same time, some other train is running through, it's noisy, you might have difficulties understanding it. So we thought, could we generate artificial facial animations just from a speech signal that might help somebody to better understand that speech tone? And we used so-called generative adversarial networks. There are a particular type of deep neural networks that essentially have two deep neural networks that are in competition. One network is a so-called generator network. That means it takes, in our case, a still image of somebody's face plus the speech signal. And the generator then produces many images of that face with a little variation, so that if you play them, you get an animation of the face that should look like that person is talking. And then there's a discriminator network. The discriminator network takes the artificially generated sequence of images plus the actual video of the person who actually talked. And it tries to decide which of the two has been made by a computer and which of them is the real one. And so both networks are in competition. That means the generator network tries to make as good facial animations as possible to fool the discriminator network. And the discriminator network tries to identify as small features, as small ingredients as it can to find out the smallest difference to be able to see what is artificial, what is natural. Now, you see here the following two videos, one on the left side of a speaker, then another one on the right side, saying the same sentence. And one of them is the natural video. The other one is the computer animation. Yeah, so I give you the same task now as the discriminator network gets. That means you can now be the discriminator. You can decide, or you should decide, which one is the natural video and which one is the artificial one. Yeah, so one left, one right. So let me play that.
Set white by N8 soon.
Should I play it again? Or we make a vote, so who votes? What is the natural video? Who says left? Who says right? I would say there's a majority. And that's right. So the right video, that is the natural one. The left one is the computer animation. A little bit more fuzzy perhaps, but I just play it again because
Set white by N8 soon.
They certainly look both very natural. I would say, I think most people would say, you've got a question up.
I think the blinking in the left one looks unnatural. Like it's not his mouth, it's his eyes.
Well, possible. Let me check that, or let me verify.
Set white by N8 soon.
So he is blinking, but maybe a bit in a weird way, right?
In the wrong place. I don't know.
So the funny thing is, maybe this would be different in this audience, right? But the funny thing is, we did a Turing test, to do this with loads of people. Play two videos, ask which one is computer generated, which one is natural. And the answer, as you can see, is basically 50-50. So at least in this population that we tested on, people didn't reliably manage to pick out which one is which. Which is funny. Now, we thought then, well, hopefully these artificial networks help to improve speech and noise comprehension. We hadn't trained them on that, right? We had trained them on producing as natural looking videos as possible, but not specifically for the purpose of improving speech and noise comprehension. But we thought, well, let's see how that goes. Yeah? So we did with Enrico Varano, another student, we tested that. So playing these videos with speech to people, speech and noise. We chose the noise level so people understood without the videos about 50% to make it hard, right? So if it's without noise and people understand everything anyways, so that doesn't make sense. But if it's noisy, then they understood 50% without the videos. With the natural videos, they understood, I think about 70%. And funny enough, with these synthetic animations, they land right in between. So I understand maybe 60%. So for us, that was quite surprising. So good news in a way. It means these animations do help. They have meaningful information in them. but not yet as much as the natural videos. So although it looks like from the Turing test we can consciously not differentiate between them, maybe except for a very few very talented people who pick up on blinking, most of us cannot. When it comes to testing speech and noise, there's still a gap. So that's something that we want to work on further to reduce that gap. We want to improve the generation of the networks to involve speech and noise understanding using, again, audiovisual speech recognition. The funny thing is audiovisual speech recognizers, here abbreviated as AVSRs, they show a very similar behavior. So they have a certain gain. So gain means how much with the increase in performance compared to audio only. That gain is higher for the natural videos than for, it's higher for the natural videos than for the synthetic ones. So we think that if we incorporate these AVSRs as maybe instead of the discriminator networks, we can force the generator network to make videos that produce a better benefit for enhancing speech and noise comprehension. Maybe producing videos that look quite different, right? Maybe they might not look that natural anymore. Maybe they might even emphasize additional things that even maybe get us to understand yet more than with the natural videos. Let's see. That's work that's open, but I think there was a question before. Was it answered already again? Okay. So that's something that we are excited about going forward. And one question is also how much time I have actually.
Until 5:30.
5:30. Okay. Then this is a bit of time. I won't get through everything, but that's not really important. And again, please ask questions, right? So
They can do questions at the end. It's better for you to get through the slides.
okay. Then, well, right. Let me go to that. So again, we thought then, well, audiovisual is great, and again, we want to do more on that. But what are other modalities? How we can enhance speech and noise comprehension? And one of them is the tactile domain, so people feeling certain vibrations. I'll come to that in a minute. But before, we thought, well, let's look a bit at the kind of basic neurobiology. Let's try to figure that out in another way. And our way was a transcranial current stimulation, so a technique where we influence brain activity to some degree by also electrodes that are positioned on the scalpel. Now, what do we want to influence in that way? Now we want to go to a different type of neural response, namely related to speech rhythms. Speech is a complex signal, of course. There are different building blocks, phonemes, syllables, and words. And these building blocks, funny enough, happen on time scales that are quite prevalent in cortical activities. That's not much slower than what I showed you before. Yeah, no longer at 100 Hertz or 200 Hertz, but rather things, well, maybe gamma range phonemes, 30 to 50 Hertz, could also be higher. But especially these quite low frequency activities, namely related to syllables that lie in the so-called theta range, about 4 to 8 Hertz, and words that lie in the delta range, 1 to 4 Hertz. And there's a lot of work. Usha has done a lot of that. Other people as well, that these brain responses in this delta and theta frequency range kind of entrain or track speech features, basically rhythms of words and syllables when we listen to speech. So there's a correspondence between these brain activities and basically these amplitude fluctuations or these onsets of words and syllables in natural language processing. So they seem to play a role, but what exactly is that role? Well, we started looking at a computational model motivated by prior work by Alexander Hühnerfiel that built artificial neural networks using spiking networks. One network that produces gamma activity through excitatory and inhibitory populations that are coupled. Another one that produces theta activity, again, through excitatory and inhibitory populations, again coupled. Both of them receive input from speech. The theta network is slow, so it only receives kind of a single channel that's more or less like an amplitude fluctuation. The gamma network receives input from many auditory channels, so filtered into different frequency bands. And what happens then is when one now presents speech to this network, that the theta network kind of produces activity that to some degree aligns with the onset of syllables, and the output of the gamma network, if one builds a machine learning classifier, that can allow to decode the identity of the syllable. So in some sense, this network together can encode aspects of speech, first by parsing speech into the syllable boundaries, and second then by encoding syllable identity. Now, something similar might happen in the brain. We don't quite know, of course, but that's some picture that we have in mind. What we wanted to see is, can we test this, or some aspects of that, by using this technique that I mentioned already, TACS, or transcranial alternating current stimulation. So we put electrodes on people's heads in this technique, and there's a slow current flowing between them. Alternating means that goes back and forth, say in a sinusoidal manner. And it's known from animal experiments that through the sinusoidal stimulation, one can align multi-unit activity of neurons towards fire to some degree in sync with the stimulation. So it's not that the stimulation induces neural activity, it rather shuffles it to happen more or less in sync with, say, excitatory phases of the stimulation. Now, other people had actually already started working on that, namely Lars Rieke, Benedikt Zofel, actually here at CBU, working with Matt Davis, and also people in Oldenburg in groups there, to pair presenting speech with stimulating using this transcranial alternating current. And the idea being that instead of a sinusoidal signal, we use a signal based on the speech envelope, so on amplitude fluctuations, because these amplitude fluctuations, they are the ones that we think are used by the brain to do the syllable and word parsing. So by stimulating, we think we can maybe influence them, and that's in effect that we hope to see. And we did that actually. Mahmoud, who's here in the audience now working in Usha's lab, he worked on these experiments. We actually wanted to selectively look at delta and theta band. That means we took the speech envelope, but we filtered it once in the delta range, once in the theta range. And we considered different phase shifts. So all these lines shown by different colors, they represent phase shifts of that original signal, because we thought if you look at different phase shifts, then if there is an influence, the influence should show up in a somewhat sinusoidal, certainly cyclical pattern. And that's what we find for the theta band, but not for the delta band. So here on the y-axis, we measure speech comprehension. So we present people with single sentences and noise, where they understand, in this case, maybe roughly 50-something%, here also between 55 and 60%. And we stimulate at different phases. We also had then charm stimulation as additional control. And we see that for the delta band, there's no effect of the phase, so we don't see effect on comprehension. But for the theta band, we find this very clear, more or less sinusoidal effect, certainly cyclical with a phasor. And the effect is not that weak, so we go from maybe understanding 60% in the best condition to understanding just 55% in the lowest condition. So not like super high, not that we suddenly understand 20% more, but still we understand more by some significant amount. And what's interesting is also that when we use this best condition, that is in this case no phase shift, actually 0 degrees, and we compare it to having this delta stimulation with no phase shift or certain charm stimuli, basing this on unrelated speech, then we find really an improvement. So we see that we can actually improve speech and noise comprehension through that type of stimulation. Now this is interesting, I think mostly from the basic science point of view, that basically this delta stimulation that we think is linked to the rate of words didn't give anything. We think delta, also knowing other work, is more related to higher level linguistic processing. It might be quite difficult with these simple stimulations to influence this in the right manner. Delta band, theta band is linked, we think, to the syllable processing. We think that's lower level acoustic, and it looks like that's more amenable to stimulation through this kind. That means we can actually influence that in some way that we see effects on behavior. But of course, for practical purposes, having these electrodes on your scalp is not very meaningful, very practical. So we were looking for another way, for another modality to perhaps deliver a similar type of signal. And for us, another modality was the tactile domain. Why that? Well, there are quite some experiments on monkeys that show that there's actually direct input from the somatosensory system into auditory cortex. Quite surprising. We might ask ourselves, what is it for? I'm also not quite sure. But there's work from Peter Lacato's lab, for example, that show this auditory somatosensory interaction in, again, monkey brains in primary auditory cortex. And actually also, especially in this delta and theta band that we are interested in. So we were interested in seeing whether this works. So can we build tactile stimuli that maybe people initially feel over their fingers that interact with their speech and noise comprehension. There was one group in Maastricht by Lars Krieke that tried before. They tried to base tactile stimuli again on the envelope of a speech signal and then looked at different delays, but weren't really successful at finding any, behaviorally at least, any relevant effector. There's also work from Mark Fletcher in Southampton who had success in CI patients. For CI patients, again, speech and noise comprehension is an important thing. And he found the effect was particularly strong when he included training. That means when subjects experienced this combination of speech and tactile vibrations many times to have some training doing that. Now we took a somewhat different approach, or a little bit different. We thought, well, if the important thing is really the syllable parsing or enhancement, then it might make sense to go to the so-called perceptual centers of syllables. They seem to play a critical role. And so we made parsers that were located at these perceptual centers of each syllable. And we designed little parsers so people would feel like little beep, so to speak, but not in audition, but through this tactile modality. We delivered these tactile motors, or these tactile signals through these little specialized motors, quite small. We asked people to hold them between their fingertips of their thumb and index finger, and then also recorded EEG because we also wanted to see what happens neurally. Can we see also effects there? And so it turns out that yes, we see behavioral effects, also quite significant. Here are so-called Z scores. That means we normalize the speech comprehension data to have a mean of 0 and then standard deviation of 1. And we see that depending on the delay, so we also have then different delays between the stimulus, the tactile stimulus and the speech. We do certainly get either decreases or also increases in speech comprehension. Interestingly, the best prediction is again for 0 milliseconds in this case. That means that these tactile pulses happen exactly at the perceptual centers. We didn't quite expect that. So you can see here that we had included more negative delays. Negative delay means that the tactile signal happens before the auditory signal, because we thought that if the tactile signal happens earlier, that kind of sets up the brain, so to speak, or the auditory cortex in the right way. So it is then prepared for the auditory input. We don't find that, but of course, there are different delays again between the auditory and the somatosensory system. So maybe that works in the right way. And thank you. And so here in this case, for this setup, again, having the exact alignment between the vibrotactile pulses and the speech leads to the highest level of speech comprehension. Just to see what the effects really are, here again, we have the scale in percent, and we compare then the stimulation at 0 millisecond delay to audio only or a sham stimulus, and we see an increase of something like 7%. So again, it's not huge. It's not like what we see with audiovisual, but for somebody who has difficulties listening in noise, it might actually already be a quite sizable improvement. All right, then this last thing I will mention and just basically quickly go over that. We looked at these EEG recordings and did quite some analysis, again, building these models that I mentioned before, these decoders basically, like in this case, backward or forward models, predicting things like speech envelope, because that now contains this information about onsets also of syllables, computing multisensory gain by comparing what do we have when people listen to speech only, when they only experience the vibrotactile signals, and when they have both. And a second analysis that was quite similar. And we find then for these multisensory gains, similar curves depending on this delay. So again, the highest multisensory gain for no delay between the tactile signals and the speech, and otherwise less following this approximately sinusoidal pattern. So an interesting analogy. So we think that yes, this EG measure of this auditactile gain does indeed say something about behavior, although we're also sure this can be improved because that's still a quite simple analysis using the speech envelope, but it shows certainly something there is indeed related to behavior. So with that, let me jump ahead just to summarize what I've shown you. So I told you about work that we're doing on speech processing in the brain, on effects of attention, partly in the brain stem, midbrain, but also partly in the auditory cortex. How, on the one hand, we can see that from the basic science point of view, but also how we might use it in practical applications, going to smart hearing aids, trying to decode this in more or less real time. And okay, some things I didn't mention, like surprisal, but I'll show you another time. Next steps are things like, if you want to go down the practical route, improving model performances further using deep neural networks. That's something that we have partly done already, but we're doing more on that. Of course, then going to wearable devices with a few EG channels only, looking at hearing impaired subjects. That's something we started recently, working with people at the TU Dresden on cochlear implant patients and people with hearing aids, and then trying to integrate all that together. So with that, I would like to thank my group. I highlighted some of the people already who worked on that. Pierre Guimino, for example, on the auditactile system. That's my current group in Germany. Here's Alina, who worked on the MEG. Thanks to funding agencies, and of course, thank you for your attention.
Well, I'd like to thank Tobias. That was an outstanding talk. What a lot of material, and the most amazing publications all in the last 3 or 4 years. It's really a tribute. Very well done. We now have 2 minutes if anyone needs to leave, and then we'll do half an hour of questions.
Otherwise, you're stuck here forever. So you mean maybe I'll write this out.
Maybe I will say
Criticize him first. Good luck. I was going to say, can you just say all that again, please?
Okay, who would like to ask the first question?
So that's a really fascinating talk, and I've got 2 questions, but my second question kind of depends on your answer first. So where do you think the source of this attention is? Is it a cortical feedback process to brainstem structures from maybe say the thalamus, or do you think there's a general modulatory effect across the whole brain?
No, exactly. So I would think it's a top-down feedback, going back from cortex to different areas, midbrain, brainstem, really all the way back to the cochlea. So we have actually one paper out in Euro from 2021, I believe, where we look at audioacoustic emissions related to speech. Don't have the time here, but the idea is what happens in the inner ear, right? Because really this so-called afferent feedback, I mean, some fibers target a whole area of the cochlea, but other fibers also particular segments. So you could imagine a mechanism where certain frequency bands are down-regulated due to the feedback, others may be up-regulated. And so that could set up a little bit of a spectral filter, not so much at the higher harmonics of speech, then they're all unresolved, they're blended together, but at the lower harmonics, they're so-called resolved harmonics. That means they peak at different places in the cochlea and could therefore be individually regulated basically by this top-down feedback. So we tried to see if that's the case. We developed some technique to measure audioacoustic emissions, not by pure tones, as one normally does, but by such waveforms similar to this fundamental waveform, although higher harmonics, but somewhat related to that. And there we see that if one attends, then these a particular speaker, again having two speech signals at the same time, one seems to get larger of the speech audioacoustic emissions than if one doesn't attend. The effect was weak, so we now just started another project on that to refine this technique, make this kind of get more data on that. But my hypothesis is that already in the inner ear, one could have such a spectral filter, certainly due to top-down feedback, and then get more of that at other processing stages.
So then, my second question is, you mentioned noise from the EEG signal. Do you think if you had different data, for example, ECOG data from, say, temporal lobe epilepsy surgery, for example, that would probably, did you think the kind of predictive value of that data would be higher than, say, EEG data? And have you thought about that?
Certainly. So there are groups that do that, very few, because it's not that easy to get access, right, to that kind of data. There's a lab at Columbia University, Nima Meskarami, who works with epilepsy patients, for instance. And you certainly then don't get these brainstem responses, of course, because that's cortical, but you get then cortical responses, and of course very strong ones, right? Like much less noisy, much better attention decoding for obvious reasons, right? No question. We want to look at that as well, also from the viewpoint, for example, of looking at source localization. So we are in contact with a lab in Erlangen that does epilepsy research and does also stereotactile EEG, so really have deep electrodes inserted into different areas of the brain. They have data that could be good for us to use, where patients are monitored 24/7 for a few days. The EEG or SEEG is recorded. And also at the same time, there's a microphone in the video, so one sees them, one has the knowledge of what they hear, but it's not greatly aligned. So I didn't mention that, right? But for the sum of work, you need, especially for the brainstem work at these high frequencies, you need very precise alignment between the neural signal and, say, the audio signal. And in their data, in their questions, that wasn't important. A few seconds delay, even, or 100 milliseconds, a few hundred milliseconds didn't matter. So we want to do this again. We are now working with them to change the setup or add to the setup so we can make this precise alignment. And then I think when we then get data with this precise alignment, that's also going to be very rich on.
Thank you.
Alex.
Hello, thank you so much for the talk. I hope this isn't too cheeky as a question, but I'd really like to hear about the results that you have for surprisal. Can you tell us briefly something about them?
So let me go back to that. So that's that has to do with the surprisal then, I guess, right? Like language prediction, so predicting sequences of words. Like now ChatGPT, we can again get it in this model as well. And we computed that surprisal at the time ChatGPT was yet available. We used some other DNNs to make that. We also computed something called precision, inverse of entropy. So entropy is basically related to how certain we are in making a certain prediction. So entropy would be the inverse again. So precision means if we are pretty certain, we have a high precision, otherwise a low precision. And then we build these models where we have, again, speech. People listen to speech. and we quantify that through different predictors. Word onset would be a very simple one, just capturing kind of low-level onset information. Word frequency, that's frequency in a large corpus of text, how frequent a certain word is, and then surprisal and also this precision variable. And we do get responses to all, also to the interaction of surprisal and precision. So word onsets like that, surprisal mostly at lags of maybe 400, 500 milliseconds. So a little bit like the N400, it looks topographically a little bit different, but sort of in line with that. People have found similar things before. Jonathan Simon, for example, with MEG on so-called phoneme surprisal, where he looked not at words, but individual phonemes. And again, it's how surprising a phoneme is given the phonemes that were presented beforehand. And here we find that on the word level, we check that when using a foreign language, Dutch in this case, for English-speaking participants, and then the surprisal effects here are absent. Interestingly, the word frequency effects are there, which is funny. So I think people still get something in Dutch that they pick out on statistics, but they don't get the surprisal because they don't understand the meaning.
Yes, please.
I also wanted to thank you for a very interesting talk. This is such a fascinating area to be doing work in. I wanted to sort of back up and also kind of go off your question. So, barring the possibility of having electrodes directly in your brain in epilepsy patients to get incredibly clean signals, I wanted to know if you could discuss some of the barriers to getting more reliable results, for instance, for the decoding stuff you showed at the beginning when you were comparing, for instance, brainstem responses to begin with between attended and unattended voices. And it looked like almost, 70% of the people you could get pretty clear responses where you had a stronger signal in the attended, in the brainstem for the attended voice compared to the non-attended voice. But are there technical ways in order to implement this? If you wanted to implement this kind of thing in a hearing aid, you needed to work for everyone. Are there ways you can think of from a technical perspective or other perspective of trying to get at that 30% that you could detect the attention in?
Yeah, it's a good question. So I think when we look at this attention decoding accuracy, then I showed you these graphs where, say, we look at between 1 second of data to 30 seconds of data, that example, and something like maybe 60-70%, depending on how long data you feed in. But that's, of course, also an average, right? So that's a population average. That means there are some subjects where the accuracy is maybe 80-90%, and others where it's basically 50% or chance level. So it's an important point that you mentioned, and I don't have too good an answer other than, I guess, trying to get more data, of course, right? Also from the individual subjects. So one funny effect is that if that was available, if we had these devices, they would also, of course, collect data all the time. So suddenly you would also have loads more data. Now the data amount that we have is not big. So for deep learning, it's a very small amount of data. So if we compare it to speech processing or vision, where people have, billions of hours of speech or so, or videos, we have here maybe an hour of EEG recording with different conditions for 20 subjects. That is nothing compared to this audio and speech processing. So hope is a little bit that by, on the one hand, combining existing data between different groups that work on that, we can improve on these algorithms. Ultimately, another answer to this problem might indeed also be implants. So this project in Dresden that I mentioned, where we work with the CI patients and hearing aid users, one of the aims is actually also to develop an implantable hearing aid that sits sort of somewhere in the middle ear, is fully implantable. And if that was available and people would use it, it might not be that far-fetched to also implant something that measures brain signals, at least from the surface of the cortex. Maybe not inside, but somewhere from the surface. And that certainly would also be a game changer for that. But so far, I think, having more data, still trying to maybe also having smarter positioning, seeing fitting things to anatomy in participants, can hopefully still boost that a bit up. The other thing that I should mention is that maybe we also don't think necessarily we need 100% attention decoding accuracy, because we anyways wouldn't want like a hard filter in the sense that you would ideally only hear that particular target voice, because people certainly will switch attention. So they should also have the freedom to still hear different sources, but say preferentially one. So we think of the whole thing later being more a bit like a soft filter, where we then enhance one voice a bit more, but not exclusively, and then look at when people switch that they still have that ability, and we can also track it on.
You mentioned or conclude that you can decode attention from the brainstem response. And I can think of sort of two ways in which attention might affect your response, the response which you record. So one of them is that there's something cortical going on, and that modulates the brainstem activity, and I think that's what uses it. But there could also be, what you might be doing is recording a combination of the cortical and the brainstem response. And you're showing a maximum latency of about 7 milliseconds. But that's, clearly, whichever one of those two things is true, there's something with a longer latency going on.
Okay.
Yeah. So I'm wondering how you can distinguish between those two, because it could be that, even if the cortical response is smaller, the main latency might come up at 7 milliseconds, but the decoding accuracy bit, the attention, which differentiates between attention, might be a more cortical response.
Yeah, so we think it's really not the case because of the ways how the responses look like. I'm just trying to find the right slides. So that would be one example of such a temporal response function for measured with EEG. And so we see here that basically at so we know now the cortical response has the maximum at somewhere like 35 milliseconds.
Mm-hmm.
And here we are basically in the noise at 35 milliseconds.
Sure, but this is the reconstruction for a single source, which is 8 milliseconds,
Well, that's a model where we feed in all kind of possible delays and reconstruct this fundamental waveform. And then we find that basically at 8 milliseconds, that's the maximal, or at that delay, we get the largest coefficients. If you look at 35 milliseconds, basically there's no significant contribution.
But if for the so this is just for reconstructing one source.
You mean one acoustic
when I'm
feature?
looking at the effective attention, So if this were happening for the effective attention, so if you managed to derive something like this, for example, for the difference between the attended or the unattended source, if it's a top-down, if that thing is coming from top-down, then you've got to have something with a longer latency.
Okay.
And so regardless of whether you're measuring a cortical response with a longer latency or something which has gone up and then back down, it's still going to have a longer latency. So I don't think you can distinguish those two hypotheses just by saying, it's got a short latency, because something longer must be going on either way.
Okay, so, well, there's something longer going on, of course, I agree. This is basically, how should I explain that? So, if you would start listening to speech, then of course 8 milliseconds later, you would not yet have the attention effect, right? I'm not claiming that, because that's not possible. What we measure is basically a response that happens constantly 8 milliseconds after the speech, and we average over minutes to see it, right? So what we think is that you start listening to these two competing talkers, you focus on one, then maybe, 100 milliseconds later, 200 milliseconds later, 300 milliseconds later, something like that, the attention effect kicks in, influences through top-down feedback, this earlier response that still consistently will show up at this 8 millisecond delay. Only it's influenced later, of course, right? It's not influenced immediately 8 milliseconds after starting the whole thing, but maybe a few hundred milliseconds later. But its intrinsic delay with respect to that acoustic feature, this fundamental waveform, is 8 milliseconds. And the intrinsic delay of the cortical response to the same feature is 35 milliseconds. And these are quite distinct in time. That's how we can differentiate them. But it certainly happens, it certainly kicks in after a longer time, no question.
But the cortical response could kick in a longer time, and you might be still recording that rather than the control signal. Anyway.
Well, so my point is, with this analysis, it's quite clear. Maybe I can show you something else that we did with MEG, although it's also funny, they're quite distinct. Let me just get to that, because it's also something we did. If we've got the time, I don't know, but I think we have half an hour quickly. Because we just published, well, it's an archive we just put another paper on archive with MEG and long MEG recordings, because we wanted to see both. So to be honest with you, with the MEG attention, ideally we would have liked to see the subcortical contribution as well. But the subcortical contribution is awfully difficult to measure with MEG. So that's a bit unfortunate. But here we have loads of MEG data for speech and quiet. So no attention, just speech and quiet. And we see then we do source reconstruction, and we see both the subcortical and the cortical responses. It's all not quite ideal, right? But that's how it comes out. That's this envelope modulation feature, but think of it just as one of these two acoustic features related to the fundamental frequency. And there's an early peak here, so all these things highlighted with pink are basically significant responses. So there's the early things here, these are probably side lobes, but the first peak here is again pretty much this 8 and 9 millisecond thing, so very consistent. And then later, this thing at 35, somewhere there, again, and that's then, when we do source reconstruction, that's in sensor space. When we do source reconstruction, that is then cortical, and this first thing is subcortical. And again, I think the nice thing here is in time, they are quite differentiated. If it was the case that, the cortical thing the subcortical thing was at 8 millisecond and the cortical thing at 12, and they have a big spread, then this would be difficult to disentangle. But they are quite separated in time.
Sure. I still think that if you had this cortical thing then modulating your brainstem source, then that component of the brainstem source would be delayed, because
Why don't we discuss this in the top?
We might need this, yeah. We might need some drawings.
So there was a question at the front here.
Hi, can I ask a question?
Sure.
Yeah, sure. Very nice talk. Thank you very much. And I have a question which also might relate to a bit about Usha's study. So, like Usha is researching on how children are understanding language, and especially for the education process, how is it evolving? And what I'm thinking about is actually for autistic children. Like for autistic children, actually some of them can understand language, but probably just don't know how to speak or something, or somebody cannot. So I'm wondering, because you are like researching on decoding speech from the EEG signals. So I'm wondering if there's a way, for example, for the children to listening like some sentences and record their EEG signals and then decode back to recognize whether these autistic children do understand it or not.
Yeah, this could be very interesting. So I think the surprise that I just mentioned briefly, thanks to your question, that could be one excellent marker for that, because if you don't see the surprisal response, like say for the Dutch speech, then you certainly know that this child hasn't understood the meaning. So I don't know enough about autism to say how strong that effect would be. I could imagine that they still understand something, maybe it's a more gradual thing. But I think it's a very interesting suggestion. I could totally see that. And again, I think the surprisal might be a very consistent marker and a very good marker for that. The drawback is that to measure that surprisal, you also need quite some amount of data. So maybe 10 minutes of data, maybe ideally more. Because also a weak signal. It sits basically, I mean, these acoustic features like speech envelope or word onset are a lot stronger. And then these additional modulations through attention or say that surprisal are weaker, they are measurable, but you need enough data to capture them.
If there's not enough data, then the surprisal might not be that significant, so it's hard to analyze it.
Then you are in the noise and you wouldn't know, you don't see it, but then you don't know, is it because it's not there or because I don't have enough data? No.
Yeah, and I think the other point is that people with autism are often very literal in how they interpret sentences. So you should probably have a control condition where you have to understand, say, irony or some higher level feature which also contributes to speech comprehension, and then compare the two. That would be very interesting. Anyway, I think Brian had a question.
In the work you did with two talkers and you attend to one or the other, I assume that those had roughly equal sound levels, right? But in a realistic situation, if you've enhanced one voice so you've got maybe one is 6 to 10 dB higher than the other, which a person with a cochlear implant might need to have a chance, then that will make it much more difficult to switch to the previously unattended talker if their voice is really 6 to 10 dB down on the one you've been listening to. it may take ages to switch over because you've got to overcome that 6 to 10 dB sort of disadvantage in latching onto the new talker.
Yeah, that is absolutely right. So I don't have a good answer. So, as I said, we are starting now to work with CI patients, and I learned from working with these people in Dresden in this case, worked with them, how difficult it is for CI patients to even do the selective attention experiment that we're running in the first place. Because for them, any, say, similar distracting speaker level is a huge distraction. And even more so than if these levels differ. So to be honest, we are starting to look at that, but I don't have a good answer, right? So it might be that this is not quite feasible. We will see. Yeah, exactly. We want to figure out, I think, first now, what are issues with artifacts in these CIs? How can we eliminate those? How can we get to similar attention decoding accuracies as the ones that we see in these healthy people? But you're absolutely right. The next step would then be to look at what happens when these speakers have different levels, what happens when this thing is dynamic. We still want to make this. Our goal is to make a demo where we have then CI patients with a closed loop system where not yet quite really the implant, but say over loudspeakers, the level of the two speakers that they receive will be adjusted, and we pick up on these changes, and we will probably make our life a bit easier in the sense that we don't change the intensities by too much, but we still would like to make it such that we see a behavioral outcome. So the CI patients in these questions that we ask have behavioral accuracies of maybe something like 70%, which is in a way good for us because it means they understand a lot, but not everything. So there's room for improvement. So we think from the behavioral viewpoint, we are good, at a good location basically where we can see advances. But yeah, I'm also curious. So hopefully in 2 or 3 years, I can give you some data. Let's see in which direction it goes, where we have data on these questions are.
Yes, please.
Thank you. I wanted to ask a question about your work on syllable rate tracking in beta band. And you talked about perceptual centers, which I assume is the nucleus onset, or is that what you meant by that?
So it's basically where people put the weight of the syllable, more or less the vowel.
Yeah, vowel onset, nucleus onset.
Well, it's the rise time of the
Yes.
vowel. The rise time of the vowel, so it depends how long the vowel is. But it's...
Okay, right, so the rise times. The rise time obviously is a change in amplitude as well, by definition. So I was just wondering, that's very strong perceptual cue. So do you think that it's predominantly a neurophysiological response that you're measuring, or is it actually entrainment? So is it, you have an expectation and that's met or not, or is it just the physiological response to something that is perceptually very salient?
It's a very good question. So I think it's not even necessarily related to these P centers, but also to things like speech envelope or so. So that's really an ongoing discussion, actually. Okay, so you're aware of that. I'm glad. But for the others, right, there's an ongoing discussion. Are there endogenous brain oscillations in the delta theta band that are becoming entrained? So there are any ways that they latch on to these rhythms, or do we induce activity by the stimulation? And so my viewpoint is a bit agnostic, I have to admit, that with the, maybe going in favor of the oscillation hypothesis is the work that we did with TSES, because that certainly comes from a framework of having endogenous oscillations that then through the stimulation become entrained. Now, there might be other ways to interpret it as well, right? So I'm not saying it's necessarily true. There's a bit of work that came out recently. I think Benedikt Zöfel published a paper lately also about evidence also to do with TSES actually. And then in kind of after effects, seeing something that looks like oscillations. We have a bit of work in that as well that was published just this year actually, Journal of Cognitive Neuroscience, where we look also at presenting little tactile pulses paired with syllables at different delays, and then also looking at after effects. Actually, different. We first presented pulses to set up a rhythm, and then at different times after the last pulse, presented syllables, had a syllable discrimination task, and it turned out that how well people could discriminate it depended on the time, and in somewhat sinusoidal fashion. or damped sinusoidal oscillation. So my impression is that there is some evidence emerging for the oscillation hypothesis, but not yet totally conclusive probably.
I was wondering whether actually your delay manipulation could inform that potentially, because you said at 0 milliseconds it was the best enhancement. Sorry, I'm not sure if I'm using the correct language, but so I haven't worked it out for myself yet, but I was wondering whether that can actually maybe give us an idea and where we would expect the enhancement based on either side of the discussion.
I think it's difficult with that type of experiment because there we used running speed, and in that running speed, you do have these rhythms coming from the stimulus itself. So then again, it's not clear of any effect, say like this one, whether what you're seeing. Okay, here we see something like sinusoidal modulations. We certainly wouldn't expect them to carry on forever, right? We would expect now if we go to more negative delays or more positive delays, that this evens out, becomes damped, but in this middle range, it looks like a damped oscillation. I don't think that in itself is evidence for an actual oscillation going on. That just mimics an autocorrelation of a certain signal that you're looking at with itself. So because it's ongoing, so I think the critical experiments are really to try to set up an ongoing or train an ongoing oscillation if it existed, and then look after the simulation, however it works, has ended, is that still persisting at least for a certain number of cycles to see is there something, because that wouldn't happen if it was just all induced now.
Okay, I think we should stop with the questions because he's been talking for half